So yes, I want to get through lecture 2 today, and depending on time, we can start lecture 3. We'll just see where we are in terms of timing, and also depending on questions and chat. So what I would like to do is just kind of refresh your memory on what we specifically spoken about in terms of restriction endonucleases, to make sure to really understand some key information. So we talked the last time about the fact that restriction endonucleases are these enzymes that are used in bacteria to protect themselves from invading or foreign DNA. And the restriction enzymes that we studied, endonucleases, they're endonuclease because they cut within the DNA. And they're also specific, they also identify specific DNA sequences and cut only at those particular sequences, right? Not anywhere else. So these were just a list of some of the commercially available restriction enzymes, and many of these are commercially available from a company where you can just purchase them. And they have different types of recognition sequences that they will recognize. Where the arrow is located, that's where they're actually going to cut the DNA between these two nucleotides. They can recognize 4 base pairs, 6 base pairs, or 8 base pairs. And some of these sequences are palindromic. And you guys were clear about what palindromic means, right?
Yes.
Yes, okay. So yeah, so for those of you who are not sure, again, it's reading the sequence the same forward and reverse, right? So you're doing A, G, C, T, A, G, C, T, right? So this is the reverse complement that you're reading. And this is kind of special to restriction enzymes. The other thing that I want to make sure that you guys are aware of is that you can have 2 enzymes that are identical in the sense that they recognize different sites in the same sequence. And these are called heteroschizomers. So one example that is listed here is SAL1 and SMAL1, okay? So you can notice both of these, sorry, this arrow is not correct. So SMAL1 and XMA1. So you notice that the sequence for SMAL1 is CCC-GGG, and it's the same for XMA1. However, the cleavage site is different, right? So SMAL1 would cleave here while XMA1 will cleave here. So they're recognizing the same sequence, but cutting at different sites within those sequences. And then you have enzymes that are called isoschizomers, which they recognize the same site and they cut within the same location between the same 2 nucleotides. And this is just an example here. And this has been updated as well in the lecture that I posted last night for lecture 2, what we will be going over today. So this is important for you to know. The other thing that I wanted to clarify was that, you know, because enzymes can recognize these different nucleotides, right? Either 4 base pair sequence, 6 base pair sequence, or an 8 base pair sequence, you will have different frequency in terms of how often they cut within the DNA, right? So if you're an enzyme that recognizes a 4 base pair sequence, you will cut more often than if it was a 6 base pairs or 6 base pair or 8 base pair sequence. Any idea why that might be? You guys want to guess?
Is it because the 4 base pair is more likely to show up because it's a smaller combination?
Exactly, right? It's a smaller combination, and also because, you know, if you think of a sequence of a 4 base pair, right, it will occur, we have 4 bases in DNA, right? So it will occur once every 4 to the 4th base pairs, right? So 4 nucleotides, 4 chances of it happening. So you will cut every about 256 base pair if you're a 4 base pair cutter. So it'll be much more frequent in the genome, as you mentioned, right? Compared to if it was 6 base pairs or 8 base pairs, right? Again, you have 4 possible combination, right? A, T, C, or G, and how the sequence that's recognized, right? So 4 to the 6, that's every 4,000 base pairs for a 6 base pair cutter. And 8 base pair is a little bit more rare because you need to, it will cut every 65,000 base pairs. So sequences such as, or enzymes such as NOT1, which is recognizes 8 base pair, is often considered a rare restriction enzyme or a rare cutter because it just cuts at less frequency than others. So that's something that I also wanted you guys to keep in mind. And the other thing before we continue with the lecture is I mentioned that you can have 3 different versions once you use the restriction enzymes. And you can, restriction enzymes, once they cleave the DNA, they can leave either what we refer to as a 5' overhang, right? So this 5' is overhanging, or bottom complementary strand, the 5' is overhanging, right, as you can see here. You can also cleave where you have 3' overhangs, which we refer to, or you can have blunt end cuts, right, where it cuts directly at both locations on both strands of DNA, so it's just a clean, straight cut. And, you know, these are enzymes that were identified in bacteria. It's a way for the bacteria to prevent foreign DNA from invading its genome and causing any kind of detrimental effects to the bacteria. So the bacteria have these restriction enzymes that helps it to protect against these invading viruses. So foreign DNA, it will digest it. But it is recognizing DNA. So how is it able to prevent these enzymes from actually cutting and cleaving its own DNA, right? Because they have DNA in the bacteria. So how do you distinguish between foreign DNA versus your own DNA? Because these restriction enzymes are present and also the restriction sites are present within the bacteria. So what bacteria have is a system, what they refer to as a restriction modification system, in that these restriction enzymes are often paired or mostly paired with metallases, okay? So metallases are enzymes that add a metal group to particular bases within the DNA. So, and once this metal group has been added to the DNA, it cannot be cleaved by these restriction endonucleases, okay? So this is normally if you have a foreign DNA or what that's coming into the genome, there's no metal group. So these metallases add these metal groups. So this will be cleaved by that restriction enzyme. However, in bacteria, these sites, these restriction enzymes are present with these metallases, and the DNA is metallated at these restriction sites within the bacteria. So the endonucleases cannot cleave if there's a metal group added to this location. That's really how bacteria is able to distinguish between invading DNA versus its own DNA and not cleaved and not chewed out, okay? So because bacteria replicate, and you'll hear more about DNA replication as we talk about eukaryotic gene expression when we're talking about changes in chromatin structure and modifications that can be made to the DNA. And because DNA gets replicated, right? So you need to replicate your DNA to survive, right? Cell division. Whenever the DNA replicates, right, you have a double-stranded DNA. And those 2 strands will separate, and you have a new daughter strand that will be synthesized or made, okay? And that's the one to the bottom that you're seeing here. The top strand, which is the parental strand, right, the original strand, both of those have metal groups added to it. The new strand doesn't. During DNA replication in bacteria, you need to kind of maintain this methylated state so that these enzymes don't randomly start chewing up your DNA, right? So even though when you replicate the DNA, you only have 1 strand, that 1 strand is sufficient to prevent cleavage of that DNA by those endonucleases. DNA replicates semi-conservatively. So this is always going to be the case. You're always going to have 1 strand, the parental strand, having at least 1 metal group to that DNA. Okay, so this what we call hemi, because it's not full, so it's just partial, right? So just 1. This hemi-methylated DNA is enough to protect against cleavage by endonucleases. So what happens is then the genome, the bacteria recognizes, oh, you know, we have a double-stranded DNA, but it's hemimethylated. So we need to get this methylated and add an additional methyl group to the strand that's not, that does not contain the methyl group, right? And it reinforces it. So you start with double-stranded DNA, go through replication, you're in a hemimethylated state, so only one strand, one of the parental strands have the the methyl group added to it. And then methylase comes in and added to the other group. So this is protected for the rest of the cell cycle that's going on. So it's, you know, restricting its DNA from being digested or chewed up or destroyed, and also making sure that this continues on throughout cell division so that they, you know, they can survive. So this was a technology that was developed in bacteria. So what scientists wanted to do is figure out a way where we can actually use this natural kind of immune response from bacteria for molecular biology, right? For understanding how genes functions. And in I would say 20, 30 years ago, you know, you were interested in studying a gene, you know, you need to clone the gene. Say, because these restriction endonucleases can be isolated from the bacterial genome and be applied to any nucleic acid that you have, they were able to use this, the ability of this restriction endonucleases to just cleave DNA that's not, you know, necessarily methylated, to create vectors or plasmid, recombinant vectors or plasmid to really study gene function and to understand cloning and all these type of purposes. So Boyer and Cohen, you know, decided to just do a very simple experiment to understand how can we use these restriction endonucleases to create a recombinant molecule, right? So a recombinant DNA. And as I mentioned the last time, you know, recombinant DNA is the combination of two different sources of DNA or genetic material. So they use this restriction enzyme, which is usually referred to as EcoRI1. Some people might say EcoR1, but it's really EcoRI1 that we often refer to this enzyme. And imagine that you have two plasmids, right? So these plasmids are circular plasmid that's coming from the bacteria. And you can cleave with EcoRI1 in both of these plasmids, two different plasmids. If you cleave in these two different plasmids, right, you should just have one fragment, right? So that's clear, right? So each plasmid you digest with EcoRI1 right here, right? It will cleave it. And you really should have a kind of a linear fragment that now have two sticky ends, right? They both have an overhang because that's how EcoRI1 cleaves, right? It creates this overhang. And then you want to mix them together, so you'll mix them together in a test tube, and you're gonna have different pools within that test tube. You will have some plasmids that will just anneal back together, right? So it will be like this, all purple. You'll have some that's all yellow, but then you're gonna have this recombinant plasmids where it's half purple, half yellow, right? So this is the different options that will happen if you were to mix these two plasmids together with just the one cutting with EcoRI1. So once these two pieces are stuck together, you will need an enzyme to seal them back together, okay? And the enzyme that's used for sealing two parts of DNA or nucleic acids together is referred to as DNA ligase, okay? It's a DNA because it recognizes DNA, and ligase meaning that it ligates it together, okay? And it's able to do that because in the natural sense, you have a double-stranded DNA, you're creating a cleavage in this case, right? You're gonna have one end that's gonna have a free hydroxyl group, right? So this is usually the 3' end as from directionality of DNA. And you also have this phosphate, which is often the 5' phosphate, which you have. And you need to seal, now you need to seal this break or this nick in the DNA. The enzyme that does that is DNA ligase, but it cannot do this on its own, right? It needs the help of ATP. Very early on in the first lecture, I mentioned that ATP is kind of the energy storage molecule within the cell, okay? And that's the same for also NAD, which is also another molecule that helps to store energy. So in this case, the ATP, which is adenine triphosphate, right? So there's 3 phosphate groups that are attached to this adenine, attaches to this ligase. When it attaches to the ligase and reacts with the ligase, that ligase then becomes activated. So now it's an activated enzyme, okay? Being an activated enzyme, it's able to donate a phosphate to this phosphate, this 5' phosphate in the DNA sequence. By donating this phosphate to the phosphate here in the DNA sequence, this free phosphate group in the DNA sequence, it creates a really highly charged diphosphate, okay? So this diphosphate that's then present is really high energy. It's really a high energy diphosphate. In order to seal this nick, so this that you have a diphosphate is storing all this energy, highly energetic. You need to say you want to seal this nick, right? So you want to create a phosphodiester bond with this OH that's present. This nucleotide that's attached to this OH. And what happens is that then you need to break this bond. So you're creating this double phosphate group or this diphosphate. Then this needs to break, right? Because that breakage of that high energy diphosphate bond is what really helps to seal this nick, okay? So you're starting, you have your DNA ligase, you need to activate it, it creates a diphosphate. The energy that's gonna come from releasing that diphosphate is what really helps to seal this nick within the DNA. And that's really essentially how ligase works to seal any kind of breakage in the DNA. You'll see it's in, we all have DNA ligase within our genome because we will have cleavage or breaks, DNA breaks that needs to be repaired and sealed back together. So with the help of endonucleases and ligase, you can fuse 2 different sources of DNA or molecules together using these in combination in test tube. So then once you're able to create these molecules, you need to get them into the cell, right? So you're creating a recombinant molecule using either restriction enzymes and this ligase to cleave them together. You need to really get this information into the cell, okay? Because it's molecular biology, you're creating a molecule, you wanna be able to produce more of it, right, within the cell. And the way that you do that is through transformation. You remember Griffith experiment, right, where you guys remember what the Griffith experiment was actually? Anyone? Yeah, the rats, exactly, right? So exactly, so where you had the rats and you also had the 2 different types of bacteria, right? The virulent and the non-virulent, where you were infiltrating the mice, exactly. So I see a lot of, I see all the information coming in the chat, exactly, right? So you need to transform this information into the cell, and there's 2 commonly used methods for transforming E. coli, okay? And E. coli, of course, is the host that we use within the lab, and I'll tell you a little bit how that's a little bit different from the E. coli you might think that that usually can infect us to cause bacterial diseases. It's a little bit modified version of that. But the one that we use in the lab is a version of this E. coli strain. And you need to get your plasmid or your DNA into the cell, okay? So, exactly, yes. So someone asked if we use a non-virulent strain of bacteria in the lab. Yes, we do. And I'll show you how that's been modified, I think in some of the few slides coming up, of what we had to modify to be able to do that, yeah? So there's 2 ways to get into the cell. The very easy and fast way that we normally do in the lab is using this chemical method. You guys recall that I told you that DNA is very negatively charged. So what you do is that you mix this DNA with a calcium solution. Calcium is highly positively charged, right? So by mixing these 2 together, And also the membrane, the cell membrane is also negatively charged. So this positively charged solution enclosing your negatively charged DNA kind of forces it to become in proximity with the negatively charged membrane. So, right, so opposite attracts, right? So it pulls it towards the membrane and then creates a little bit of holes in the membrane from just that force, yeah? Some holes in the membrane, and you can shuttle the DNA across the cell membrane, and it's in the cell, okay? And this is the common way that you use it in the lab. It takes a few hours. There's many different optimization, of course, that has been done so that you can do this easily now that we're doing it, but this is the method that's most commonly used. You can also use an electroporation method, which is very similar to the chemical method, except instead of using calcium, you're using an electric charge. And you're using an electric charge to really just force this negatively charged DNA across this membrane. Okay, so it's a pulse of electricity that so you'll have your culture in a particular device, and you will apply an electrical current, and it will just force the DNA across the membrane by creating pores in the membrane. Okay, so these are the two ways that you can transform E. coli. And this is not, of course, not the only ways to transform any type of cell. There are many different ways that you can transform a cell, depending on the organism. So the question is, which one might cause more stress on the organism? So if not done correctly, you can imagine that the electroporation method will be a little bit more stressful for the organism. So whenever you're doing the electroporation method, it's really like a really brief pulse of electricity that you're applying to the solution. And then you immediately put it into a cold environment for them to recover. Okay, so you have to kind of take these precautions if you're going to do the electroporation method, just to make sure that the cells are, don't die because of this electrical pulse. So that's why it's more commonly, the chemical method is just more commonly used, and it's just a little bit easier, and they're just as equally efficient. Yeah. So you can transform different types of cells. Not all cells are competent, like E. coli, right? So some cells they have, for instance, in plants, right, you have a cell wall. It's almost impossible to get anything across a cell wall, right? So you need to find different ways to get your DNA across the different barriers that might exist for different cells, okay? So these are all different methods that you can use to transform a cell. Of course, bacteria, which we just talked about, using calcium. You can also do this with fungi, so things like mushrooms. You can also, some animal cells can also be transformed using this calcium method as well. Electroporation works well for all organisms, right? Because regardless of what barrier exists, you can just force everything across the cell with an electric pulse, okay? Microinjection using a needle. This is often done in both animal and plant cells, so you inject directly into the cell. You can also bombard with DNA-coated microprojectiles, so some beads. So you can have some metal beads that you coat with your DNA, and then you use kind of a gun, which we call a gene gun, and actually shoot the organism that you're working with, and it will just transfer it into the cell. In terms of plants, and particularly most of the transformation we use in plants uses this special type of bacteria that adheres to the cell and injects its DNA into the cell. So this is the method that's used most commonly, I would say, to transform plant cells, is Agrobacterium tumefaciens. And you can also use it for other systems as well. Lipozyme is a little bit unique to animal cells. So on that type of technique here is, you have your DNA, you have these liposomal molecules that's full of lipids, and they're able to encase their positively charged, you encase your negatively charged DNA, and it kind of shuttles it across the membrane for you, and then your DNA gets in. So that's kind of a method that's really unique, I would say, mostly to plant cells. Okay, so you can use many different ways to get your DNA across the membrane or across the cell, into the cell. But in order to get them across, not only do you need to consider how are you going to get them across these different cell walls or cell membranes, etc. You need to have a special type of molecule for this to work, right? So you need to be able to create a construct, what we call, and we call these constructs cloning vectors, right? Because in bacteria cannot replicate linear DNA, right? The bacteria plasmid of itself is circular. So if you're going to put something into bacteria, it needs to be circular, okay? So your piece of DNA, if you were to cut a piece of your DNA and say, okay, I want to clone this into bacteria, you need to put it into a vector to create a circular molecule, okay? And you need, so there's two main types of vectors. So there's plasmids. Again, these are primarily coming from bacteria, right? Small circular bacterial plasmids or replicons because they can replicate easily. And there's another type of vector that we use depending on the purpose. It's not as common as plasmids, and I'll tell you a little bit more about that later. What's the purpose? What purpose we mostly use viruses as cloning vectors. And on those vectors, you need to have very special features, right? You need to be able to have things that tell the bacteria, I want you to do this with this particular plasmid, okay? And some of the features that are important, right? And you need to remember this because these are kind of the questions that you would see on an exam to, explain what are some of the important characteristics of a cloning vector, right? And you'll see this later on as well in other classes. Or if you work in the lab, you need to know what features are present on your vectors because there's many, many, many of them, and they have so many different things that some of them might not be relevant to you, others might be relevant to you. But for the most basic information that needs to be present on a cloning vector to be able to use it as a cloning vector, it needs to be able to stably replicate in the host. Right? And that means it needs to have an origin of replication. Where does replication start? Right? So that needs to be present on your vector. You need to also be able to easily introduce these vectors into a host. Most of what we talked about is introducing them into bacteria. You can also use these similar vectors to introduce into yeast cells. And if you're using a plasmid, you will transform that plasmid using one of the multiple methods, but more likely using the chemical method we talked about, which is the easiest, or by infection. And you will get back to that Hershey and Chase experiment where we talked about how you can infect using viruses a little bit later. You also need to have a marker on your vector, right? You need to be able to say, okay, I have my vector in my bacterial cell, okay? How do I know I have my vector? I did my transformation using one of these great methods. How do I know I actually have it in the cell, okay? So you need to be able to select for the cells that contain your vector because not all cells are gonna have it, right? And the way you do that is by using an antibiotic resistance gene on your vector. So this is a gene that's resistant to a particular antibiotic that's present on that cloning vector. So if you were to grow your bacterial cells on a plate containing that antibiotic, it will not die, right? It should be able to resist it and grow on that plate if it contains that vector with that antibiotic resistance gene, right? So you need to have a selection marker. You also need a place to put your DNA, right? Do you randomly put your DNA anywhere on this vector, or where do you put your piece of DNA that you want to clone, right? And so you need to have special sites on those vectors where you can actually clone your DNA. And these sites are usually referred to as a multiple cloning site, or MCS. These sites contain 10 to 20 different restriction enzyme sites that's clustered within this multiple cloning site. So you can use any of these 10 to 20 different restriction enzymes to cut your DNA and cut that plasmid and put them together, okay? So these are kind of the 4 very, very basic features that you will need to have on a cloning vector that you need if you're going to use it. That's clear? Yeah, okay. So the plasmids that we use as vectors all initially were created from a very classic series, which is called the PUC series of vector for E. coli. Do you guys have any idea why it might be called PUC? So P stands for plasmids, and UC stands for the University of California. Okay? Yeah. Okay. So someone did say, yeah, it's from the University of California. Exactly. Right. So these vectors were developed by a professor at UC Davis, and he was working on bacteria. He was also working in cloning. He's a molecular biologist. So he developed these series of vectors that can be used for cloning purposes. On these vectors, they contain a bacterial origin of replication, multiple cloning sites, and a selectable marker. We know that it's easily can be transferred into the host, so have, again, all the desirable traits that we're interested in for a vector. In addition, what's also present on this vector is a screening marker. So a screening marker is important if you want to know whether or not your piece of DNA has inserted into the region that you want it to be in, okay? And this is a way to quickly figure out not only if your transformation work, right? Because that's what your selectable marker is going to tell you, did my transformation work? What the screening marker tells you is whether or not your piece of DNA has inserted into that multiple cloning site that you want it to be in, okay? Of note, not all plasmids contain a screening marker, because now we've developed different types of plasmids and have modified these very basic classic vectors to have additional features where we can easily screen them to know whether or not our vector has taken up the piece of DNA in the right location. So this was just in the very beginning. Now things have been modified a lot more, but this is still being widely used, these screening marker types of experiments, because sometimes you don't have a choice and this is what you have to use. Yeah, so getting an understanding of this is really important. So this is an image of a classic real vector that we use in the lab, right? So PUC19, it's 2,686 base pairs. So it's a pretty small vector. Some vectors can be 10,000 or more. Someone asked, so the MCS is really the multiple cloning site, right? So this is where you're going to be putting your piece of DNA, as we mentioned back here, which you'll have. And I'll show you a little bit more detail what that is in a minute. Right, so here it is. So this is your plasmid. This is a real, true plasmid that we use all the time in the lab. It contains an antibiotic resistance gene, right? And this is for ampicillin. So it's usually written as APR, or sometimes you might see it as AMPR, right? So it tells you that this region contains that selectable marker, okay? You also have the origin of replication. You know where that origin of replication is. And you have your multiple cloning site. I told you that's a region that contains multiple restriction endonucleases sites that's present within that region. So this is your MCS. And this MCS is located within your screening marker, okay? This design needed to be this way in order for us to be able to use it as a screening marker, okay? So you can imagine that because this multiple cloning site where you're gonna put your piece of DNA is located within this screening marker, if you disrupt this screening marker by putting a piece of DNA here, then you would disrupt the function of that gene, right? So you can just imagine that. And we'll talk more about this in a minute, but just get the general sense. You have a gene and you're putting something in that gene, you will disrupt the normal function of that gene, right? I mean, this is what will happen. So the screening marker present within this vector is a beta-galactosidase gene. Beta-galactosidase gene is a gene that's present in bacteria and also it's used in other species for many different things, and also you can also find used as a reporter in yeast system for yeast experiments. But you can use this beta-galactosidase gene as a screening marker, and I'll tell you why we're able to do that. So you have the beta-galactosidase gene is called LAGZ. When we talk about bacterial transcription, you're going to become very familiar with this LAGZ. And what we have within this plasmid is not the complete beta-galactosidase gene or LAGZ gene. It's just a part of it. And it's referred to as LAGZ alpha, okay? Because it's just a part of this gene that's present in this plasmid. And within it contains this MCS. So the way that the selectable marker works is, again, you have your recombinant plasmid. Here's the plasmid, you will digest with a restriction enzyme, right? So it's open, right? So you have a cut. It has the ampicillin resistance gene, which is present. And you also have, it doesn't show here, but you also have an origin of replication because you have to have it, okay? So you have all of this vector present. You have your piece of DNA. You notice your piece of DNA is also digested with the same enzyme as you're cutting your vector with, right? Because you want to put them together. And you add DNA ligase, which is important for sealing them together. And now you have your plasmid. You have your plasmid, you have the resistance gene, your selectable marker. You have the region, it's inserted somewhere in your multi-cloning site where you cut with this enzyme BAMH1. You transform it into E. coli using one of your different methods into E. coli. And now you look to see whether or not your cells will grow on ampicillin, right? Because this is the way you figure out whether or not your transformation worked, okay? So in the first case, if your transformation doesn't work, would you expect your cells to grow on ampicillin? No, right? Exactly. So you wouldn't expect anything because it cannot survive on ampicillin. It doesn't have the ampicillin resistance gene present. And exactly. So then now if you transform this now plasmid and it has your insert, it will grow, right? So it will grow. This is what you expect. Doesn't grow or it grows, it has your plasmid you want. What happens if this insert didn't insert, because you guys remember in the beginning I told you, you have a cleavage with a single enzyme. You can have some of the vector it's just going to reanneal to itself, right? You're adding ligase, so there's no reason that this is not going to come back together and not take up this insert, right? And this is the situation you will have, right? Where the vector comes back together. And this happens a lot. About 50% of the time, you will have this. 50% of the time, you will have this. If you were to digest your plasmid with a single enzyme, add an insert that's digested with a single enzyme. This is reality. This is what will happen, right? Because it's by chance, right? If you add ligase, these are going to come back together. And then the other half is going to seal these with the insert together, yeah? So what would happen in this case? Would these cells grow on the on ampicillin, on a plate that contain, or a media that contains ampicillin? No? Okay. Okay. Any reason why you think it's no? Yeah, so what they lack, actually, so they will grow. The reason they will grow is because even though they don't have the insert, they still have the ampicillin resistance gene, right? So they will still grow. This becomes a problem if you're looking at a screening marker, okay? So in the case of a screening marker, you would not get the expected outcome if it's not present on the vector, okay? But for transformation, all you need to know is did your transformation work? Is do you have growth on these media containing antibiotic resistance? Yeah, and if you do, then you know, okay, your transformation work, you have a plasmid in there. You don't know whether or not the plasmid have your insert, but you know that you have a plasmid, right? So you have either of these two. You just don't know which one you have. So the way that you figure out which of those two options you have is to use the screening marker, okay? So, okay, so the question is, what is the purpose of the insert? So if you're gonna be cloning something, you need to put the insert into the vector. You wanna clone this piece of DNA. That's your goal, right? That's your end goal, right? So that's the purpose. Your end goal is, I have this piece of DNA, I wanna study it, I wanna put it into this vector, and I wanna make tons of it so I can work with it, right? So you have the plasmid that you will digest, you wanna put your insert in there, okay? When you do this reaction, putting your insert into this plasmid, it will be transformed into the bacteria. Not all of your plasmids are going to have the insert, only half of them will have it, right? And the other half will not have it, right? The other half is gonna have the plasmid, but the plasmid is by nature is just gonna seal itself back together because there's nothing that's preventing it from coming back together, right? So you're gonna have some that have your insert and some that doesn't, okay? You have to have this ampicillin resistance gene because you need it to survive in the bacteria. You need to have this ampicillin resistance gene because you need to know whether or not you've transformed your plasmid. And the only way to know that from the millions of cells that bacteria will make is by putting it on something that you can select for it, yeah? So the vector is really, this is the cloning method. You're just using the vector and the plasmid to make more of your piece of DNA. All right, that's really the goal. So just the cloning, how do we get from, I have my piece of DNA to get to where I need to be able to study it, okay? So once you have that vector containing your insert, the way you distinguish, or once you have that plasmid transform into the cell, the way you distinguish whether or not you have your insert, right? Or you have your insert, which is the recombinant transformant, or you just have an empty vector, just an empty plasmid that came back together. How do you distinguish between those two situations? So you have a plasmid, foreign DNA here, right? You can see, you can have one of these three options that will happen when you do the ligation. What we talked about in the previous slide is you have your plasmid, you see the foreign DNA is inserted right there in the multi-cloning site. That was one of the options, right? The other option was that you have, the plasmid and there's no insert. So the plasmid just came back together and sealed, right? So these are the two options we just saw previously. Both of these are gonna grow on ampicillin because this is the ampicillin resistance gene. They both contain them. In the cell, they both contain them, so they can grow on the ampicillin resistance gene, saying that they have transformed into the cell. This option, where you have the insert, but somehow, you know, this is what happens naturally, right? Somehow this insert, for whatever reason, didn't insert in that multi-cloning, the multiple cloning site, but it inserted somewhere else on the plasmid, right? Somewhere else. Yeah. These cells will also grow on ampicillin. This transformation work, they're all successful. They all have the resistance gene in this cell. Now, how do you distinguish these three scenarios where the insert is in the correct place, it's in the wrong place, or it's not there at all? Okay, and that's where the screening marker becomes important. Because now what you're screening for something that's creating a blue color versus a white color. Why a blue color versus a white color? Okay, so beta-galactosidase, usually it's an enzyme that produces a blue color when it's fully functional. Okay, and in order to determine whether or not you have this blue color, what you need to do is include a particular substrate, which is called XGAL, okay? So this substrate is important because what it does is that it hydrolyzes this beta-galactosidase and creates this blue pigment, okay? So if beta-galactosidase is there, it's functional, then you will have blue colonies being produced. IPTG is also another compound. It's really an inducer, and it really induces the production of beta-galactosidase as well. Okay, so you combine two things that helps to either make the cells blue or induce the expression of the gene, and these two will only work if that gene is functional. Okay, if that beta-galactosidase gene is functional. So in this scenario, right, you don't disrupt this beta-galactosidase gene because you insert it somewhere else, right? So you guys recall what I mentioned, that you have to disrupt this, right? The multi-cloning site is here, you put something here, you will disrupt this gene. If that insert goes somewhere else, this gene is not disrupted, it's still functional, okay? So yes, blue is bad, right? That means that your insert is not there, right? So it's somewhere else. Here, if you also have the plasmid that didn't take up the insert, this beta-galactoside gene is still functional, you'll get blue colonies. The only way you're able to distinguish when your insert is located in the right location is when it disrupts this beta-galactosidase gene. So all your colonies cannot produce the blue color. So your colonies are all white. Okay, so this is usually referred to as blue-white screening or lax selection or alpha complementation. And it's really you're screening for clones that contains your plasmid in the cell and your insert is in the right location. Yeah, so how do you separate blue from white? You plate it on a plate. This is a real plate that you have. On this plate is both XGAL, IPTG, and all your bacterial cells. So for further studies beyond this point, what you would do is take one of these white colonies and go with it, right? Because the white colony should have it. Okay, so the blue indicates, as I mentioned, either your insert is there on the plasmid, it's just not right in the right location, or it's not there at all. So your colonies are blue. If it inserts in the right location, disrupts that LAX-D gene from its normal function, then all of those colonies will be white. Okay? And then we just go from here. We will pick up, this is what we do in the lab. We will pick up the white colonies. Maybe you pick up 4 or 5 of them just to make sure you have enough, because a lot of weird things can happen in bacteria that you're not, you can't control. And then you go just continue on with the next step to get your plasmid that you want. Okay? So the next step beyond this would be to isolate that plasmid for further study. So now you need to get the plasmid back out of bacteria. You just put it in, bacteria made tons of it for you. Now you want to get it back out of bacteria. So that's kind of the next step. Okay. So how this blue-white screening work? So someone asked about the multi-cloning site. Is this use MSA or you mean MCS? Someone asked about the multiple, if you're referring to the multiple cloning site, as I mentioned, this multiple cloning site is really just a region on that plasmid that contains multiple restriction enzymes where you will cut and put your DNA in there. Okay, is that clear? So it's just a region on that plasmid containing many different restriction enzymes like you were seeing here. And you would use one of these restriction enzymes to cut the plasmid so you can put your insert in here. Okay. So this blue-white screening, the ability to do that is because you have your plasmid, you have just a part of this whole beta-galactosidase gene that's actually present, right? So you don't have the whole thing, you just have part of it, okay? So you have one part that's present here, and then the other part is present in the bacteria on the bacteria chromosome, okay? So that's the basic concept to be able to use such a system. You have to have one part of it on the plasmid and the other part within the bacteria, okay? And the reason for that is that if you have part of it, okay, you need to make sure this plasmid gets into the bacterial cell, right? Once it gets into the bacterial cell, bacteria will have its own chromosomal plasmid, which are circular, containing part of this, part of that beta-galactosidase gene. So you see it's like ZDM15, so that's just part of it. This is the alpha part, which is the other part. When it's in the bacteria, if this part is functional, meaning that this part is produced, this part is produced, they will come together. When they come together, they form this tetrameric structure, this homotetrameric structure. And homo just meaning that it's the same molecule, and tetramer just meaning that there's 4 of them. And they come together, and that is what produces a functional beta-galactosidase gene. If this is disrupted in your plasmid because you put an insert there, you make this part, but then this part will no longer be made. So then you cannot form this functional homotetramer, and then you cannot have a functional LAC-Z gene, okay? So it's kind of called alpha complementation because you're kind of complementing your normal bacterial cells with this alpha part, okay? So if the LAC-Z gene is present if it transforms correctly in the plasmid and it is not disrupted, you will have a functional LAC-Z, your cells will be blue. If you cloning worked and you inserted a fragment in this portion of your LAC-Z, it will be disrupted. When you transform it into the cell, it cannot make this structure, so you will not have blue, and that's why your cells are white. Okay? So I'm sure you're going to go over this more in your discussion sessions, and if you don't, just let me know by office hours or by email, and I'll try to clarify. But we should move on. So yeah, you have blue colonies, yeah. If you have a functional and not functional, it will be white colony. So as I mentioned, as Sheila mentioned, or asked you, what are the bacteria that we use in the lab? So the bacteria we use in the lab, again, is the similar E. coli strain that normally infects humus and causes all of these intestinal infections that we might have. But we've done a series of mutations. So all of these numbers, which you don't need to memorize, are just different modifications we made within genes in that bacteria, right? So it was initially isolated in Palo Alto. We have created mutations for some of the cell wall genes, right? Which makes it not able to colonize the intestines anymore, because that's what normally, if you get a bacterial infection or food illness, right? Colonize your intestines. And it also, so we modified it so this does not happen. And also it raises the transformation efficiency so it can be easily transformed because the cell wall is more easily susceptible to that. And then there's a variety of modifications, one of them being this LAC-Z gene. We modified it so we can use it as a screening marker. So that's the reason. And And someone said that so blue is, just to clarify again, if your plasmid does not contain the insert in the right location where you want it to be, so if it doesn't disrupt this LAC-Z version, then you will have blue colonies because your LAC-Z can still function. White is good. So white means that you've disrupted LAC-Z. That means you likely cloned your insert into the right location. So as someone mentioned, yes, blue is bad, white is good. Yeah, in this situation. So some other modifications we made again, just to make this plasmid efficient for us to use. So another modification that we need to do is it's difficult to have all plasmids and all vectors with a screening marker, okay? So if you don't have this screening marker, you know, you're gonna still get 50% of the time you have the insert. The other 50% of the time, your vectors are gonna come back together, right? So it's really a big problem for vectors without blue-white screening, and many vectors that we use on a daily basis do not contain this blue-white screening. So how do we tell from the hundreds of cells we have on the plate that are all white, right? Because there's no blue-white screening, they're all gonna be white. How do we tell which one actually have your insert or not, right? Because they're just growing on that plate because they have been transformed with your plasmid. Whether or not they have the insert, you don't know, right? I mean, you just see colonies on the plate. And you don't have this blue-white screening. How do you figure that out, right? Of course, you will screen millions. You can screen by different methods, like by PCR, which we will talk about later. But to help to reduce the vectors from coming back together, what we normally do in that case is we use an enzyme called alkaline phosphatase, okay? And what alkaline phosphatase does is that alkaline phosphatase removes the 5' phosphate groups from your vector where it cuts, right? Because remember, you're gonna have 5' and 3', 5' and 3'. So it removes these phosphate groups. Because the phosphate groups are removed and you add DNA ligase, these can't come back together. There's no phosphate to bring them together, right? So you don't get any of these plasmids coming back together, okay? Only when you add your foreign DNA, the piece of DNA you're trying to clone, those will contain the phosphate and they will be able to go into this vector and DNA ligase will seal it, okay? So this is really how you reduce having a vector that's empty versus a vector that has your insert, okay? So in this case, which was the example we showed previously, you cut with BAM-H1, you cut your insert goes into the vector. But before you do that, you want to treat it with alkaline phosphatase so that it doesn't close back up, and you can insert it in, you can add your insert in there. Okay, so that's the way you will prevent self-ligation. So if you were to get a question in the exam, how can you prevent your plasmid that you're trying to clone from coming back together, right? You would think immediately, oh, I'll just add alkaline phosphatase because that will remove the 5' phosphate groups from the vector, okay? So you'll reduce that from happening. So the other type of vector that we can use for cloning is usually referred to as phages, right? You saw phages before with Hershey and Chase, right, where they use that bacteriophage that they labeled with radioactivity to determine whether or not it's really DNA or protein that was being transformed, right? So it's the same idea. It's a different type of phage, but it's a phage that infects bacteria. And that can be used as a way to clone pieces of DNA. The great thing about bacteriophages is that it can infect much more cells. It can infect cells much more efficiently than plasmids can be transformed. And the clones, instead of having clones as colonies, what you have is you have rather plaques. So space on that, within that mixture of bacterial cells that are dead because the phage killed, the phage has died and has released all of the components within the phage. So you have a clearing on the bacteria, within the bacteria, the pool of bacteria, okay? And that's the way that you kind of screen for whether or not your transformation has worked to some extent. And this is a plate of an actual transformation with a bacteria phage, right? And what you're looking at is a plate. All of these white parts, this gray part, those are all bacteria, right? The patches where you see these circles of clearing, those are the plaques, right? Because that's where the bacteria phage has lysed and released all of its components. So there's a clearing. So your DNA and your clone DNA is within these cleared regions, not in the bacteria, okay? The process to create a phage was initially developed by Fred Blattner and colleagues, and they used a phage which we refer to as Sharon-4. And these type of phage vectors can receive large amounts of foreign DNA, so up to 20,000 base pairs of DNA. With plasmids, you can transform, you can clone a piece of DNA that's pretty large, maybe 5,000, 6,000, but trying to clone within a plasmid a piece of DNA that's 20,000 base pairs is very difficult. Yeah, I mean, it's really, really not efficient. It's very difficult. So you're putting for large pieces of DNA, so you want to sequence the entire human genome, right, which is super large, right? You want to be able to make these large chunks of vectors so that you can make more of it, okay? And in addition, what's also kind of special about these vectors is that they require a minimum size of foreign DNA to be able to be packaged into this phage particle. So if by random chance you just have a small piece of DNA that maybe broke off and tries to assemble into this package, it will not work, okay? So it's a little bit more specific as well, okay? Someone asked whether or not they're removing the phosphate groups from the plasmid OH interact to get the same way that OH and pH interact. So if you remove the phosphate groups, the two OHs will not interact. That's why the vectors cannot come back together. It's only when the insert is there providing that phosphate group, then they're able to come back together. So with cloning in this phage vector, so you have, this is what you will purchase from the company, right? You will purchase these pieces, right? Which you will need to put together to create an infectious phage, okay? So you have the two arms of this phage, and then you have these stuffer sequences when you buy it, right? So this is what you're purchasing from the company. To put your piece of DNA into that region, you need to remove this stuffer sequence, right? So this is just there so that we know that this is the region you're gonna put it into. You will cleave with an enzyme. You have your insert, which you also cleave with an enzyme, and you will put it into this phage. There's the other parts, right? So you need the protein-coated surface. You also need another kind of, I guess, like a structure, packaging system. So these are the three things that you're gonna mix together. It's gonna create this infectious phage. This is gonna infect bacteria, right? So you're gonna put it into bacteria. It's gonna replicate. This DNA is gonna replicate so much, then at some point, it will lyse this phage particle, and that's what will create this clearing on the cell, okay, on the plate, okay? There's a lot of details going into, you guys remember that the region between here is really about 16 to 20 kb. Only if you put a fragment that's 16 to 20 kb in this region can you assemble this package. If for some reason you put a shorter, maybe 4 times, because E. coli 1 is a 6 base pair cutter, right? So being a 6 base pair cutter, how often would you expect the E. coli 1 to cut within this DNA?
Isn't it like 4 to the 6?
Yeah, right. So like about 4,000 base pairs, right? So you can imagine if you really digest this fragment efficiently, you're going to have these 1, 2, 3. You're going to have 3 small fragments. These 3 small fragments cannot be assembled into this vector. So what is being shown here is that you have to digest with E. coli 1, but you only do a partial digestion. So very mild digestion so that you don't cut at all of these sites all the time, okay? So you're gonna have a mixture, even with a small amount of E. coli 1 for a short amount of time, you're gonna have some that remains this larger fragment. Some of them are gonna cut smaller, some of them might cut like here, so you have this size fragment. So you're gonna have a mixture, and only the one that's large enough will be assembled into this package, okay? And then you're able to get your phage. Once you have your phage on the plate, the way you determine whether or not your plasmid is present in that clearing on the plate is you have to do what we refer to as plaque hybridization. So you plate the bacteria and phage mixtures on the plate, incubate, you grow at 37, bacteria likes to grow at 37, yeah. And the clearings are where there's phages, where you have the lyse phage, right? Because there's replicated so much, it just bursts in the end. And now you need to get the phage and the protein, the phage DNA and the protein off of this plate. So what you use is a filter membrane, just rest it very gently on that plate, maybe for a second or 2 seconds, and then you remove that filter paper. Once you have your filter paper containing the spots and everything that's on your plate, you can then use the plasmid you know that might be radioactively labeled as well to see whether or not you can find it on that filter paper. So this is a hybridization method using something that you know to check to see whether or not it's present on this filter membrane. When we talk about southern blot, this will become a little bit more clear to you, but that's the method that they're using, okay? So in summary, the vectors we have examined so far are used primarily in the first stage of cloning, right? When you put it foreign DNA into bacteria, you get it to replicate, and then you have it, okay? This is for making any DNA, right? If anything you want to clone, right? Any part of DNA that you want to clone, you put it into bacteria, bacteria make tons of it. You cannot use bacteria for certain types of gene expression studies, right? Or for genes that might be highly expressed. So if you're studying a human gene that's highly expressed, yes, you can probably clone some of it in bacteria. You might be able to get some level of this gene being amplified, but in many cases, you would need a specialized expression vector, okay? And that's what's required. So the main function of an expression vector is to yield the product of a gene, right? Because you're looking at a gene. So expression, when you think of gene expression, you're always thinking of a gene, right? How is this gene expressed, okay? And it needs to be expressed really well. The more product you have, the better. And in order to have a lot of that gene, you need to have a very strong promoter, okay? So promoters are important for gene because they're the ones that direct gene expression, okay? The more mRNA you have, right? So you will express the gene, you will first make your mRNA. The more mRNA you have, the more mRNAs is available to be translated into a protein, right? So more mRNA, more protein you will make. So the other reason that you would need to use an expression vector is that eukaryotic genes are not always at home in bacterial cells, right? So they're not able to be processed properly in bacteria. Okay, you can make more of it, but in terms of actually studying gene function, they're not going to be able to function as well or have the same function in bacteria. The reason is E. coli cells sometimes recognize the protein products of a cloned eukaryotic gene as an outsider, and it will destroy it, right? You know, it's an invasion, right? So it will destroy it. And also bacteria doesn't have the same kind of post-translational or even post-transcriptional, as you'll see, modifications as eukaryotes do, okay? So you need to use a system that you can actually study this bacteria. And so that's where eukaryotic expression vectors are important, okay? And if you are really interested in cloning this outside of bacteria, you can use a, you can express these genes in a eukaryotic cell using one of the different transformation method and using different types of vectors, right? You can also do this in eukaryotic cells. Initial cloning will be used in E. coli, right? Because it's easy. You clone everything in E. coli, you check to make sure everything looks good. Do you have your gene sequence? Is there any mutation? Is it aligning with the promoter? And then you would put it into a eukaryotic vector and then put it into eukaryotic cells, okay? So two of the more important advantages are, again, eukaryotic proteins made in eukaryotic cells tend to be folded properly, because you will see later on as we talk about translation and post-translational modification. Protein folding is incredibly important for function. And eukaryotic proteins made in eukaryotic cell are also modified in a eukaryotic manner, because that's really important for the gene function, for it to be able to be modified the right way. So that's cloning using restriction enzymes, okay? So with the advent of being able to clone, being able to transform into the cell, we're using these screening markers to determine, do we have the plasmid we need? We're isolating our piece of DNA by restriction endonuclease. Sometimes it's not possible to isolate your PCR, your piece of DNA by restriction enzymes, either because maybe there's no restriction enzyme sites near to the region that you want to clone. So then you have to have an alternative method. And that's where, PCR or the polymerase chain reaction has really helped to revolutionize the, molecular biology and even cloning, right? So in the early 1980s, some researchers, Kary Mullis and some others, found a way to be able to isolate pieces of DNA using this technique called polymerase chain reaction. It's by far the most powerful technique that's ever been developed in, recombinant DNA biology or research in general, okay? It's an in vitro technique, meaning it's done in a test tube, right? Versus in vivo is in the live cell. It's an in vitro technique done in a test tube for the amplification of a region of DNA between two regions of a known sequence. Yeah. The advantages is that a target sequence of DNA can be amplified significantly higher, right? Billionfold in several hours. And you can use any type of molecular cloning strategies and analysis for gene expression that you want, right? So it's really, again, going off of, instead of isolating your DNA by using restriction endonucleases, now you're using PCR and saying, okay, this is what I want. I need to be able to clone this particular region. So in order to do that, so you have a piece of DNA that you want to clone. In order to have a PCR, to perform a PCR amplification, you need to have oligonucleotide primers. These primers are short, single-stranded. They're referred to as oligonucleotides because, again, they're single-stranded pieces of DNA that's short. And they're usually complementary to the regions you want to amplify, right? So the outer regions of the known sequence. You need two of them because you need to know where to start and where to stop, okay? And these primers serve as primers as well for DNA polymerase, because DNA polymerase is the enzyme that's going to be doing the replication and amplification. Okay, so they serve as a primer to tell DNA polymerase, okay, start adding nucleotides and extending and making more of this particular DNA within that region. So you get new synthesis of new DNA strands, which are complementary to your region of interest, okay? Oh, and also because you know where you're starting, right? And as I mentioned several times, and you guys probably already know this from previous classes, DNA is replicated or synthesize 5' to 3'. So your 5' region is always known. The 3' region is gonna depend on when you stop extending, right? When DNA polymerase stop extending. So that can potentially be variable. So it can be ambiguous in length, right? Because that can change a lot. So in a standard PCR reaction, right, you have your double-stranded DNA, and you say you want to amplify between these two regions. You have double-stranded orientation, again, 5' to 3', 5' to 3', the other strand. So you guys recall how high you need to heat this double-stranded DNA for it to be denatured?
Like 90 degrees?
Yeah, right, 90 and above, right? So you separate the strands, you break the bonds, you have 2 single-stranded molecules. You add your primer. Remember, you're going to have 2 primers telling you where to start, where you're going to end. Primers are always written in 5' to 3' orientation. This just have to be this way because DNA polymerase can only extend at this 3' end, okay? It will extend here, so it'll go in this direction, it'll extend here, and it will go in this direction. So you will have your 2 molecules after your first round of PCR. You're going to have 2 double-stranded molecules now coming from this one single strand encompassing this region. Okay? And in this typical reaction cocktail, right, we talked about the fact that you have your DNA that you want to clone, right? That could be anything, right? You decide what your DNA is, whether it's a gene, whether it's a random piece of DNA, whatever. You have 2 primers, as I mentioned. In this case, they can be 20, they can also be much shorter, but here we said 20 to 30 nucleotides long, 2 primers, right? One telling you where to start, one telling you where to end. You need a thermal stable DNA polymerase. I'll explain why that is in a minute. You also need the nucleotides. You need to provide, remember, this is all in the test tube. This is not in your cells, so this is not available. It's a test tube. You need to provide the nucleotides so that the nucleotides can be attached to and extend your DNA that you're trying to make. Sometimes you need magnesium chloride because it helps the DNA polymerase to function more efficiently. You'll have a range of different temperatures, right? Depending on, to some extent, the polymerase and also your primers, right? So you will have a cycle, so it's called a polymerase chain reaction. So you're using polymerase, the chain reaction is repeating this many, many different times to a certain extent, okay? The first step in your PCR reaction is denaturing. You need to denature your DNA so that your primers can anneal to that single-stranded DNA. Your primer is single-stranded, you need another single-stranded DNA so that your primers can anneal. And DNA polymerase can start extending that annealed that region where the primer is annealed, okay? Denaturation temperature, 94 degrees Celsius, to some extent depend on the polymerase, but for the most part, most polymerase can still function even at these high temperatures. So that means that the polymerase has to be really thermostable, and you'll see that that's the case. Annealing temperature is going to be depending on your primer, how your composition of your AT and CG within your primer sequence. All of that is going to be important. And the extension is also going to depend on your polymerase, right? So the optimal activity for many polymerase, meaning how the temperature it can function efficiently at, is usually 72 degrees Celsius for most polymerases, right? And that is just how quickly it's able to add nucleotides to the piece of DNA. So this is a reaction that you will include. You adjust it again, depending on what you're doing, your piece of DNA, the length of your primer, the GC content of your primer, and the polymerase that you're using, okay? And this is, again, just summarizing what that will look like, right? So you can go through these steps. You have denaturation. You're adding, again, primer is going to be attached. Remember, you're adding your nucleotides. They come in and they're going to be added with your polymerase and extend this piece of DNA. After the first round of PCR, right, you have a double-stranded DNA. you have 2 double-stranded DNA, and the cycle goes on. So you repeat this usually for 20 to 35 cycles to be able to have billions of copies of this piece of DNA. You need to have billions of copies because you also need to be able to visualize it, right? So having billions of copies make it much easier for you to be able to clone and also visualize. So that is kind of important for a DNA amplification step. And I'll stop in a minute. I just need to, if you don't mind, 2 or 3 more minutes, I can just get to a good stop point instead of just right here. And these, you know, back in the day, because you have to be able to use these different temperatures. When this was developed in the 1980s, right, early 1980s, we didn't have the machinery to be able to put our PCR reaction in a test tube and put it into a machine and say, okay, do 95 for this time, do 50 to 60 for this time, and then do 72 for this time. That didn't exist at that time. So instead, what they had was they had these water baths, yeah? And each water bath was at a different temperature. So this was 94, this was 50 to 60, and this was 72. So then someone, a really great undergrad like you guys, stood there, put your samples at 94 for however many minutes you need, then moved it over to the other one. Someone mentioned that's how you did it. I'm amazed people are still doing this now, but yeah, it worked. And this is how it worked back then, right? But we got lazier, we got advanced, we advanced in technology, of course, and we're always looking to make things more efficient. So now we have a range of, this is stuff at 2010, of course, the 2020s, of course, we're gonna have more newer technologies. Now we have a range of PCR machines, which is really small. You can literally, some of them are even smaller now. These are still pretty large compared to what we can have now in the lab. And you really have a machine where you can put your solution in there and just tell it, okay, I want you to do 94 degrees Celsius for 5 minutes. I need you to do this for another minute. And repeat this 20 times. And that's it. You put it in, you walk away, you come back, it's done. Okay? So that is the easy way to do it now. And it's like unbelievable how efficient this is. It is what we're using now for sequencing, genomic sequencing. As you'll see, everything was just changed because of being able to do PCR and how that actual amplification works. So I will stop there. You do have some exercises, extra credit exercises that will be available for you to do. And I would say give it your best shot. So there's an exercise that will be available after this class that helps you to think about primer design. I'm trying to see whether or not I should release this. I will release it even though we haven't gotten through all of the details of primer design, which I had hoped to today. And you guys just give it your best shot. Yeah, and we can discuss this in class, and I can give you another example to practice on next week or maybe in the next class, okay? So I'll have that release. You will get a notification saying it's available. You have 20 minutes. There are 2 questions related to restriction in the nucleuses and also related to primer design. So you can just try to do your best for showing the PCR, the restriction, and the nucleus you should be able to do because we covered it. But PCR primer design, some of you might know it and some of you might be able to easily do it, but I feel like I still need to get through what you should think about when you're thinking of primer design. Yeah, and the assignment is going to be available. It will be due, like the last time, it'll be due at 11:30 p.m. tonight. It'll be open from when you start. You have 20 minutes. So if you decide that you want to start at 11 p.m. tonight, you'll still have 20 minutes. Whether or not you start now, you still have 20 minutes to do. Yeah. And again, I'll take into account, depending on how everyone does, I'll have another example where you can actually be able to work on it better later on. Okay. So I will, we're almost done with hopefully with lecture 2, and then there's lecture 3. So again, don't worry too much about how far we've gotten. your exam will cover always up to where we've stopped. I don't expect you to know material we haven't covered. So, but these are really important steps that you need to know, that you will need to know for the future, for your future, you know, especially regardless of where you end up. Yeah. Okay, so I'll just stop there. If you have any questions, let me know. I'll stop recording so you guys can comfortably